Hello, everyone. Welcome to today's webinar. I'm Jeff Huber, contributing editor at Chemical and Engineering News, and I will be moderating today's event. This webinar is titled Introduction into Fragment-Based Drug Discovery and is being sponsored by WuXi AppTec. CNEN works with sponsors to identify topics of interest and value to CNEN's audience and consistent with CNEN's mission to provide news and analysis of the chemistry enterprise in a timely, accurate, and balanced fashion. During the webinar, you can adjust the size of the slides on your screen by grabbing the lower right corner with your mouse. If you need technical assistance, please look at the Help widget at the bottom of the screen or type your query into the Q&A box. If you are disconnected during the webcast, please log in again according to the instructions you received earlier. You are encouraged to contribute to the success of this webinar by asking questions at any time during the presentation through the Q&A box on your screen. The questions will be answered at the end of the presentation, and as your moderator, I will be posing as many as time permits. Please note that CNEN does not endorse any company, products, or services that may be mentioned in the webinars, and that each webinar will be archived at CNEN online after the live webcast. The presentation today is being sponsored by WuXi AppTec. WuXi AppTec is a global company that provides a broad portfolio of R&D and manufacturing services that enable companies in the pharmaceutical, biotech, and medical device industries to advance discoveries and deliver groundbreaking treatments to patients. And during today's presentation, we will hear from Dr. Andreas Schoop, Head of Medicinal Chemistry at WuXi AppTec. Dr. Schoop studied chemistry at the University of Münster in Germany. After receiving his PhD, Dr. Schoop completed a postdoc with Barry Troust at Stanford University, before then moving into medicinal chemistry as a lab leader with Bayer in 1994. In 2001, Dr. Schoop joined Boehringer Ingelheim in Vienna and contributed in multiple late-stage kinase lead discovery projects to deliver compounds for the clinic in oncology. Dr. Schoop joined GPC Biotech in Munich in 2007 to lead drug discovery teams in oncology and became head of medicinal chemistry with the CRO company Proteus in 2012 for integrated drug discovery projects. In 2022, he joined the hit identification platform HITS of WuXi AppTec as head of medicinal chemistry. Located in Munich at the Cyrillic site, Dr. Schoop supports clients in validating and optimizing hits from either of the two hit generation technologies, which are fragment-based drug discovery and DNA-encoded libraries. And during the presentation, we will also hear from Dr. Maureen Jarabek-Williamson, Head of Biophysics and Screening at the hit identification platform HITS at WuXi AppTec. Dr. Jarabek-Williamson studied biology in Dusseldorf, Germany, and completed his PhD at Cincinnati Children's Hospital in Cincinnati, Ohio, focused on the development of gene therapeutic applications involving MGMT protein and studying DNA damage repair of alkylating agents in hematopoietic stem cells. After receiving his PhD, Dr. Jarabek-Williamson joined Nanotemper Technology and was involved in the development of microscale thermophoresis, a powerful technology for studying biomolecular interactions. Since 2017, Dr. Jarabek-Williamson has led the biophysics and screening department at HITS, supporting many WuXi clients in biophysical and FS services to enable their drug discovery programs. And with that, I will now hand things over to our first speaker, Dr. Schoop.
So, my name is Andreas Schoop. I'm the head of medicinal chemistry at Grelux, WuXi AppTec, and I'm going to present you the introduction to fragment-based drug discovery. And this is only the part that will cover the technology of fragment-based drug discovery, where my colleague will cover then later technology on the screening for fragment-based screening. To start with the fragment-based drug discovery, I think it's very important to understand what we're looking at in terms of chemical space. For the understanding of fragment-based drug discovery, I think it's important to talk about the size of libraries and the chemical space behind those libraries. Usually we have different hit generation technologies available. This is high throughput screening, this can be virtual screening, or this can be DEL screening. And with these kind of screens, we are looking at screens with 1 million or even 1 billion of compounds. While if we do fragment-based screening, we usually have only 1,000 or a couple of thousands of compounds. And I think it's important to understand why that is and why the fragment-based drug discovery or fragment-based screening is using much smaller libraries. We need to ask the question of the relationship between the molecular weight of fragments versus drug-like compounds and the chemical space that lies behind that. And for that, I have the next slide. And on this slide, I'm showing you the relationship of how the growth of the chemical space is dependent on the number of heavy atoms. This work goes actually back to two different groups. There is the group of Jean-Louis Raymond, who has enumerated the chemical space of smaller libraries up to the heavyweight atoms of 17 atoms. And that information was then taken on by another group of Alexandre Vonek, who developed a function to that. And this function is shown here in this equation, and this is a logarithmic function. And if we now use that function and look at the different molecular weight boundaries of 300 versus the molecular weight boundary of 500, we can calculate that drug-like space for 500 is 16 log units larger than the fragment space. And with that knowledge, it is quite easy to understand that a fragment library can cover the chemical space much better than a larger HTS library that covers the molecular weight 500 space. There are another more advantages. That is probably one of the biggest advantages of the fragment-based screening, that we have a higher penetration of the chemical space with these fragments. But fragments can also fit small pockets just a lot better than larger compounds would do. And so they have a higher probability to bind, while complex lead-like molecules undergo quite often just unfavorable interactions because they're just bigger. There's also the finding that with fragment screening, we usually get higher hit rates in the range of 3 to 10%. And that can be used as an indicator for a target and for the druggability, whether a target is druggable. Another advantage is the fact that once you start with a smaller fragment and still have to grow a couple of 100 Dalton, there is still a lot of room to maneuver your chemistry in and to optimize multiple parameters. And that can be considered being a quite good advantage. And that also includes the management of IP space, because you can actually manage and maneuver your chemistry into new molecular space. On the other hand, of course, there are some requirements that need to be considered to make fragment-based screening and fragment-based drug discovery work. First of all, there needs to be clear that the affinity that we will find with fragments is more in the micromolar to millimolar range. And because we have to do some more optimization, we are dependent on some optimization techniques and what we call usually the optimization metrics. It's also understood that we are working at high concentrations of the compound, and therefore we need to have high solubilities for these compounds. Also, at these high concentrations, the screening needs to be suitable for these high concentrations. That's why we usually use either NMR, SPR, MST, ITC, or X-ray for these kind of screenings. Also, what we need to consider is for the optimization, because there is still so much chemical space we need to penetrate to get to a drug-like molecule, is that we need to be guided by structure information. And so that becomes an essential part of fragment-based drug discovery, that we have crystal structure or NMR structure information available. And finally, also there's one more point that needs to be considered. Since those libraries are so small, fragments are mostly singletons, and so it makes a lot of sense to add medicinal chemistry at an early point into the project to support the optimization of these compounds and to support the crystallization of these fragments. The HIT-S fragment library collection that we have consists of 3,100 fragments. They are compiled the way that they have a high diversity, of course, are synthetically accessible, and they follow the rule of 3, meaning that they have a molecular weight cutoff of 300, hydrogen bond donor of 3, hydrogen bond acceptor cutoff at 3, and C log P below 3. We've also looked at reactive compounds and eliminated those, and we have filtered for classical medchem filters. And finally, also, those compounds were curated in the way that they have been visually inspected and curated in the way that only attractive and classical medchem compounds are in that library. Another parameter that is important is that the number of rotated bonds are kind of limited, and that's why we like to have ring systems in those molecules. And you can see here in the bottom the distribution of the ring counts, and there is hardly or only little 2% of compounds with 0 rings, but we have 1 ring, 2 ring, or even 3 ring systems within the library. On the right-hand side, you get a flavor of how those compounds look like. This is a principal component analysis, and this is already with these red dots, the second generation of compounds, where we try to maximize the diversity and get into a larger chemical space. So once we have screened and identified hits, we go into a fragment hit triaging process. And I've shown you here the process on the left-hand side. So we usually assume that we have a hit rate of 5% with 3,000 fragments in the library. We assume that we get something like 150 primary hits, which are then validated by using an orthogonal assay. And in addition, if possible, we would also look with a functional assay, so that we get very quickly down to maybe just 100 or 50 compounds that then go into crystallography. Now, as I've mentioned before, crystallography is key for this technology. And so we need to make every effort to enhance the chances with crystallography. So for crystallography, we usually try to get an apo structure so that we can do soaking. But in some cases, we are also fine with co-crystal structures so that we generate just co-crystals. But still then, this is a difficult process, and sometimes just the numbers of fragments that get crystallized are quite low. And what we can do is that we usually can support that process by doing what we call the hit expansion, by adding medicinal chemistry to the project and either do catalog analogging or synthesize additional compounds. And that has 2 advantages. One advantage is that with catalog analogging, we're getting multiple compounds of the same cluster. And there is something like bundle concept in crystallography, and that crystallography gets multiple chances for almost the same compound to crystallize a fragment. And secondly, if we generate more compounds, we also get the chance to even improve the affinity. And with the affinity, there is just the lesson learned that the best affinity compounds usually have a higher chance to get crystallized. So that is the reason why we like to add medicinal chemistry very early in the process and support the hit identification before compounds even get crystallized. So once we have compounds or fragments that have been crystallized, we go into the process of the fragment evolution and fragment optimization. And there are multiple ways to do this. Technically, from medicinal chemistry, there are 3 ways to do this. There is what we call the fragment linking, there is fragment merging, and there is fragment growing. Here's an example of the fragment linking, where for the example of the methyltransferase DOT1L, 2 hits have been identified in proximity, but at different binding sites. And what has been done here is that a linker has been used to link those 2 fragments to generate this compound 4. Well, that compound 4 was then further optimized to generate compound 7, and that compound 7 has been crystallized, and the crystal structure shows that the compound actually adopts the conformation that was assumed. and was planned before. This is a technique that is very much straightforward on the one hand side, but also quite a difficult strategy because it requires a linker technology that is usually just working for very flexible linkers, and for that then sometimes giving very flexible molecules, which is not always desired. On the next slide is the fragment merging strategy. Here is an example from the Whitehouse group on the tRNA methyltransferase, where two compounds or two fragments have been found, fragment 16 and fragment 20. And with the fragment merging, there is no linker needed. Essentially, the scaffold or the pharmacophores are added together in a kind of molecular hybridization way. A new scaffold is generated. So on the right-hand side, you can see that scaffold 16 and 20 in an overlay, nicely overlaid, but now the boronic acid is swapped and exchanged to the amino pyrazole, and the compound 23 is generated. The pyrazole that is now connected to the indole, and that is understood as a merging. In this case, it actually did improve the affinity. And the third approach is the fragment growing, and just staying with this example of the tRNA methyltransferase, and so this compound 23 is now just taken and additional moieties or groups are added. So going from 23 to 25, the cyano group is added, and moving to compound 26F, the additional methylpyridine is added, and then finally there is the pyrrolidine added, and in that way, with the grown molecule, the affinity is improving step by step. That's understood as the fragment growing. Well, what's important for the fragment growing is that the molecular weight that is added does actually give enough potency, and that we will end with a molecular weight at or below 500, but still have a good potency. And so for fragment-based drug discovery, the concept of ligand efficiency as a metric has been established. And just to give you a definition for that, the ligand efficiency is the free binding energy per heavy atom of a ligand. And it is just important because fragments are usually not so potent, and we need to do an optimization of 4 to 6 log units in potency. Using the equation for delta G, it's quite easy to calculate the ligand efficiency. And here I've given you a table of different ligand efficiency depending on the number of heavy atoms for an affinity of 100 micromolar. And so you can see that at 100 micromolar, a ligand with 23 heavy atoms comes out with a ligand efficiency of 0.24. And if you look at maybe just the molecular weight of 200, the same affinity would translate to a ligand efficiency of 0.36. On the right side, I'm also giving you some more examples of actually drug-like molecules. Drugs, lenalidomide and rivaroxaban are two of the best-selling drugs. The lenalidomide actually is smaller and almost fragment-like, but it has a ligand efficiency of 0.40. Rivaroxaban is a molecule with 29 heavy atoms and a quite nice potency of 0.4 nanomolar. And that translates again into a ligand efficiency at 0.44. And looking at this, I think it's important to understand that conceptually, we want to use ligand efficiency as a guiding parameter. And if we take the general rule of thumb for medicinal chemistry that a drug-like compound would or should be in a single-digit nanomolar range and at 5 molecular weight at maximum, I've put this kind of spot into this activity molecular weight plot. And if you do the calculation, such a compound comes out with a ligand efficiency of 0.33. And so all what we need to do is to guide by monitoring the ligand efficiency from a fragment hit that might be less potent but small in molecular weight to that ligand efficiency and to that potency. And the observation is that it is actually quite good to start at 0.3 ligand efficiency or above that. And that would be kind of the preferred in green. I give an example that ligand efficiency of 0.45, for example, could be a good starting point. Sometimes starting at a lower ligand efficiency is possible, but for sure that is more difficult. But in some cases, that could still work, but you would want to achieve a ligand efficiency of 0.3 quite quickly. In the next example, I'm going back to the example I've been showing before, the tRNA methyltransferase example. And I've calculated the ligand efficiency for every single compound here. And I've put these compounds into the plot where we're plotting the IC50 or the PIC50 versus the molecular weight. And here we can see that we actually did achieve what we wanted to do. The IC50 is improving and we are ending up at a very potent compound and a quite nice ligand efficiency of 0.34. And we do see that trend that the IC50 is improving. If we look at the ligand efficiency in general, we did start at a ligand efficiency of 0.41 and end up at the ligand efficiency of 0.34. And so with the graph on the right-hand side, I want to show that this is a general trend we used to see, that we are usually losing on ligand efficiency. And that is one of the reasons why if you pick a fragment to start with, you would want to pick something that is at least 0.3 or even higher than that. Beyond the metrics of ligand efficiency, there is quite a plethora of different metrics, and some of them might be useful, some of them might be less useful. Here I've just picked a few of them. I personally always like to work with ligand efficiency. There's something that is quite similar, that's the binding efficiency index. And then there are metrics that do not guide in respect to the molecular weight, but that guide in terms of the lipophilicity. And so there is what's called the lipophilic efficiency, which is monitoring the distance between the PIC50 and the C-log-P. And you will want to create there quite nice gap of maybe 5 or 6 log units. And so that is a number you would want to increase in your optimization. There are other lipophilic metrics like the ligand efficiency dependent lipophilicity, the LELP, or there are guidance parameters that include the polar surface area. Now, there is for sure quite some experience within the field which ones to use. My preference is always to use ligand efficiency and lipophilic efficiency. But over the time, there has also been some changes in the field, at least if you follow the recent reviews in terms of what kind of techniques to use or what kind of libraries to use. And so the newest trends seem to be that the molecular weight cutoff criteria seems to go down to 250 in molecular weight. The rule of 3 has been added in terms of that the number of rotational bonds should be smaller than 3, that also polar surface area just from the start should be as small as 60 or below. On the other hand, people have been looking for maybe even larger libraries, and there are vendors that offer fragment libraries of up to 20,000 compounds. Other new trends are the fact that we do see covalent fragment libraries being used. We do see that fragment screening is used in the combination with DEL. Once you have identified a fragment, you can use that to design your DEL library. And most recent, we also see that fragment screening is used for RNA targets. Now, within WuXi, we offer that service for fragment-based drug discovery, and we plan these kind of projects. We usually plan for a 12-month period, or maybe even more. Here for simplification, we are just looking at the project, but have cut out the preparation of the protein. And here with month 1, we are starting with the assay development. Then there is the screening done and the hit expansion by MedChem to support the crystallization of the fragment. And that's done in the first 6 months. And once we do achieve crystal structures of fragments, we move into months 7 to 12, where we then really do the fragment evolution, as I've shown you, using these different techniques, and support that again with a lot of crystal structures. And we are quite flexible to add all kinds of additional assays or activities that are required for the project. And we can do that because Of course, HIT-S is just part of WuXi Biology, and WuXi Biology is part of the WuXi AppTec company, and we have internal resources that can be used within WuXi Biology for all kinds of assays. And of course, we can also add the WuXi chemistry for the synthesis of these compounds so that we can do all these activities in-house. I'm just looking at the structure of HIT-S itself. HIT-S is globally present in 4 locations. We are situated in Munich with the company Crelux. We have in China a site in Shanghai as well as in Suzhou. And we do have a site in Boston. And fragment-based drug discovery is performed depending on what is needed in the project, but it's either performed in Munich or in Shanghai. And with this, I would like to close my part and hand over to my colleague for the second part. Thank you very much.
My name is Moran Jarabek, and I'm leading the Biophysics and Screening Department in WuXi HIT-S. I'm very glad to introduce you in the following 15 minutes how WuXi HIT-S biophysical platform plays a central role in enabling FBDD projects in WuXi HIT-S. WuXi HIT-S' goal is to support our partners in offering an integrated solution, including protein production, FBDD, DEL, biophysical and biochemical, as well as structural biology services. Today, I will focus on our biophysical assay and screening services supporting FBDD. A successful FBDD program requires different skills as protein design and production, biophysical fragment screening and biophysical heat characterization, structure determination, and of course medchem support, which we have heard in the presentation before. All those expertise are available in HIT-S. In our department in WuXi HIT-S, we have developed a very broad toolbox to enable many different drug discovery programs. We are using different methods to solve different challenges. For studying binding kinetic, we typically use SPR and BLI. MST tricks are widely used for studying binding affinities in free solution. Both technology measure interaction in a steady state. Measurement can be performed using purified recombinant proteins, as well as under purification-free conditions. Protein integrity and thermal stability and aggregation are measured by nano-DSF and DLS. Using ITC, calorimetry can be used to study binding energetics. In HIT-S, 48K, Dianthus X, monolith and automated, 7 manual MST devices, 4 Prometheus are all being used to support many drug discovery projects of our partners, and all those methods are very crucial for fragment-based drug discovery. In the following slide, you can see how biophysical methods are being used to support different stages of drug discovery. In the beginning of the drug discovery program, it is important to characterize the target protein. Here we typically perform nano-DSF and DLS measurement to ensure that the protein is correctly folded in the assay buffer. In nano-DSF, we can test as well the impact of positive control or substrates on protein thermal stability. In addition, MST and SPR are being used from the very beginning to confirm binding of the target protein to the tool compound. During hit-to-lead stage, SPR and MST are very powerful technology to identify hits. The identified hits can be then further validated by different biophysical technologies. To make a long story short, biophysics is required during each stage of drug discovery. We typically use SPR and MST to screen our fragment collection, containing about 3,000 fragments. I am a big fan to perform a feasibility study using both methods during assay setup, before selecting which technology should be used for primary screening. Regarding throughput, all 3 technology as SPR, MST, and TRIC, as well as the new speckle shift technology, are working perfect for fragment screening, and we can measure up to a few thousand compounds in less than 1 week. It is important to say that in the past we observed that sometimes some targets work better with a certain technology, Therefore, a feasibility study is very important. But it's important to say as well that for many cases, we can feel pretty lucky, and we're glad to see that different methods work for multiple targets. So as we establish the assay with two different technology, we already establish not only the screening technology, we can directly establish the orthogonal method for the hit validation as well. Here on this slide, we can see an example of an FBDD integrated drug discovery program. Every project always starts with carefully design of the protein construct, depending on the used technology. For MST, we would like to have an His tag, for Esper an Havi tag, and the X-ray team typically prefer an untagged protein. All of this need to take into consideration before we initiate the program. After a few weeks, the protein is ready and can be analyzed by NanoDSF. Here we test the impact of different buffers and additives on protein thermal stability. In addition, binding to the positive control need to be performed as well. After confirming that the protein quality is sufficient, then we typically establish binding assay using MST and SPR, and the primary screening is then initiated at a single compound concentration of 500 micromolar. The determined binders and weak binders are then further analyzed in a KD ranking screening. The most promising binders will be then soaked into an hyperprotein crystals. The KD information, together with structural information, can be used by our MedChem team and enable lead optimization and fragment evolution. Human sting was one of the first MST-based fragment screening we have performed in WuXi Hit-S. On the left, we can see an example how the tool compound CGAMP interacts with fluorescence-labeled sting. The determined KD correlates nicely with literature value of 10 nanomolar. After the assay was established, we have performed a primary screening using a single compound concentration in duplicates. We see in red the non-binders, in blue binders, and in green potential binders. Finally, we have performed KD ranking screening using the dianthus, which can be seen on the right. To sum up, in this fragment screening, we have screened 3,100 fragments in MST. We have confirmed KD ranking 66 hits with KD between 10 and 500 micromolar. The hits were characterized in addition using NanoDSF and SPR. 9 out of 11 promising MST hits were confirmed in ligand-based NMR. One bottleneck in biophysics are often challenging, cost-intensive, and time-consuming assay setups. Last year, we have launched a ready-to-go assay platform in WuXi Hit-S. The turnover time is very short, since no protein production and assay setup are necessary anymore. Here you can see some of the available ready-to-go targets, and the available assay as a 3 ligase as Celebron DDB1, VHL complex, as well as inflammation targets like NLRP3, and other important drug targets as KRAS, PIK3CA, and Werner helicase are right now very current important drug targets. All of these ready-to-go targets are fragment screening ready as well, since all biophysical assays are already established, the protein is available, and a fragment screening against this target using our fragment library or any other fragment library can be initiated anytime. In this slide, we can see an example how the ready-to-go assay for Werner helicase looks like. On the left, we can see NanoDSF data demonstrating that the tool compound, in this case, bind ATP gamma S. And upon binding of ATP gamma S to the protein, we can see strongly increasing protein thermal stability. In MST and SPR, we have determined the binding affinity between ATP gamma S and the protein with a determined KD in the micromolar range. KRAS is another example of a very popular ready-to-go assay. Here you can see NanoDSF measurement for KRAS in the presence of BI2852, as well as MST and SPR binding data. In addition, we have established a functional nucleotide exchange assay using MANGDP. All those assays can be used for an efficient compound profiling as well as to initiate a fragment screening. Finally, we can see one of our mostly studied S3 ligases, Celebron DDB1. The fluorescently labeled Celebron DDB1 complex bind the molecular glue CC885 with a KD of 32 nanomolar. In the presence of CC885, we have measured a protein-protein interaction with a KD of 90 nanomolar between GSPT1 and Celebron. In the bottom, you can see a cooperativity experiment demonstrating that in the presence of the target protein GSPT1, there is a 40-fold increase in the binding affinity of Celebron DDB1 to CC85. Similar experiments can be performed using other 3 ligases, and this allows, of course, an in-depth characterization of binary and ternary complex formation of bifunctional small molecules. I hope you have enjoyed the presentation, and both, we look forward to starting the live session.
All right. Thank you so much for both of your presentations, and thank you audience members for submitting questions during those presentations. We've got some of those questions here right now. We're going to get to them. If you haven't had a chance to submit a question and you have one, no worries, you can still do that, and we'll still be able to pose them. So let's start here. Let's see. This first question asks, which target classes have you successfully worked on in the past using SPR or MST?
I will take this question. So we have established in general about 250 different protein targets using MST and SPR in the last few years. So we have worked on very different protein classes like helicases, TPases, kinases, as well as even some membrane protein. 3 ligase complexes are right now very hot topic. So therefore very diverse target classes. So it's always depends on the protein quality, and if we can produce a protein which is correctly folded, we put a lot of efforts on production of high protein quality. We run a lot of protein QC, and if after the initial state we can end with a protein which is functional and correctly folded, we'll typically be able to establish a biophysical assay on it. Can be MST, can be SPR, and can be another technology as well.
Our next question is about timing. They ask, when would you typically initiate the structural biology work?
Yeah, I can take that question. I think it makes sense to actually do that as early as possible. Actually, we start with protein production, and we already plan different protein constructs for the assay setup, as Moran has laid out, that the protein quality is important, but also to consider which protein constructs will be used for the crystallography, and then actually start as early as possible with the crystallography work.
Another question asks, what are the advantages of FBDD compared to other screening methods, and how about compared to HTS?
Yeah, I can also take that question. And I saw one question that was not only asking for advantages, but also for the drawbacks of fragment-based drug discovery. And so that goes back to the chemical space and the understanding that with the smaller fragments, we are looking at a smaller chemical space, and for that we are, even with less compounds, we have a better penetration of that chemical space. And the understanding is that it is easier to identify new chemical matter or enter new chemical space. And that can be a huge advantage. We have seen that in the past, that especially in crowded areas, for instance, in the kinase field where there's a lot of population and competition, where it's difficult to find new scaffolds, that fragment-based drug discovery can actually be an advantage to identify something new that you can then also manage your IP better. So that's the advantage. On the other hand, in terms of time frame, the disadvantage of the fragment-based drug discovery is the fact that you have to go through this fragment evolution process, which will cost you just additional time and, of course, also additional resources. So in the end, it might look like a cheap entry, but if you go through an optimization of additional resources with biology and chemistry, it's not much cheaper than maybe an HTS screen, and it just requires this additional time. And as I've shown, we're looking at time frames where we then get to really single-digit nanomolar potencies. That takes us something like 15 months. And so that needs to be considered right from the beginning.
Yeah, I see that there was a question that came in here about the time frame for fragment-based drug discovery, and an additional question they had with that was, what is the average range of the hit rate from fragment-based drug discovery screenings?
Yes, so from the biophysical screening, we typically see hit rate between 2 and 8%. Exactly, and all of this, of course, when we initiate structure biology work, only some of those we will typically see that we get structures. Depends on the target. It's between 5 and 20%, I would say, and that is typically a good starting point for a medchem project that Andreas is typically will be happy to initiate.
And I feel like your last couple of answers have touched on this question that I'm about to ask that was submitted. Somebody asked, when is it appropriate to use fragment-based drug discovery as the primary screening method compared with other technologies?
Yeah, maybe I should take that question. So what we see usually, I think especially in big pharma, there's kind of the standard approach that you start with HTS screening, but HTS does not always deliver sufficient hits or quality hits. And so as kind of a secondary approach, fragment-based drug discovery can actually sometimes deliver better hits or hits that HTS cannot deliver. Of course, there are other technologies too, but I do believe that fragment-based drug discovery is one of the more advanced screening technologies that comes head-to-head maybe to HTS. But of course, the one limitation, of course, is always that you need some structure information, and so I think that's the prerequisite, and that would be also important to make your judgment that you get the structure information, and if not, probably that's not the right choice to move into fragment-based drug discovery.
Another question that came in asks, do you go for single compounds or mixtures?
Yeah, I can take those. So that depends on the assay. For reversible collection, we typically go for a single compound concentrations, and meanwhile using MST or Dianthus or SPR, running primary screening of about 3,000 fragments takes 2 to 3 days, the measurement time, so that's completely reasonable. If we think on covalent fragment screening by mass spec, we typically pull 5 fragments together.
Another question that came in asks, do you need very high resolution structures for successful fragment screenings?
Yeah, maybe I
can take those. Yes.
Question.
Yes, Andreas, go on.
Or do you want, Oren? Yes.
No, Andreas,
so
go on.
For my understanding is, well, the question is definition of high resolution. Of course, we like a higher resolution better. So it depends, but probably you need at least some resolution, I would guess somewhere between, or up to 2.0. Well, and it's even difficult to say, because in the end you need to be able to really be sure that your electron density you see in the crystallography poses your fragment, and there is only one pose or one confirmation that you can do the interpretation of this electron density, and there's no ambiguity about that, because you're starting then with that position of the fragment and growing that fragment into the pockets that are available. And if that pose is not correct, then all your hypotheses and your future chemistry will not work. And so, yes, it matters, it is important, but it's probably not any limitation on to say at which point does it actually work or not. It's probably more the question of how sure is the expert in crystallography to make sure that the pose of the fragment is unambiguously correct.
Another question that came in asks, do you have any experience with a covalent fragment screening, and how would you screen those?
Yes, so we observed in a very clear increase in interest in performing covalent fragment screening using lysine or cysteine reactive warheads. And so the traditional way how to do it would be to do it using mass spec. So that's something we are doing by measuring compound pools. But recently we collected excellent experience using MST for covalent fragment screening of very relative big libraries. And the big advantage in MST that we would pick the covalent and the non-covalent fragments. And we always, after running a covalent fragment screening by MST, we confirm that the covalent are non-reversible covalent bound to the target protein using mass spec.
Another question that came in asks, can you comment on data correlations between different assays based on your experience?
So if the assay are set up correctly, we see a relative good correlation between SPR, MST, and ITC. If you think on thermal shift assay, that depends, of course, if the compound upon binding affect protein thermal stability. So in case of compound affect protein thermal stability, this compound you should be able to see using MST or SPR. But we saw in different cases that, especially if we speak about micromolar binders or fragments, you will not always see any effect on protein thermal stability, but you still can determine that this compound binds the target protein. We, especially for our fragment screening approach, we always highly recommend to orthogonally validate the fragment hits. So our validated hits are always orthogonal validated.
This next question asks for kind of a specific compound. They say if you have a compound that has around 500 molecular molars out of fragment screen, is it something you still think is good? And if so, what biophysical assay would you recommend to test its binding?
So as you may saw during the presentation, so we screen at a single compound concentration of 500 micromolar, and then we typically run KD ranking by MST or by SPR. And 500 micromolar binder are already relative weak, even for fragment screening. So we see in many cases that let's say our best is, if you're lucky, you can even determine binder with KD in the low micromolar range that go up to binding affinity of 100, 200 micromolar. With KD of 500 micromolar or weaker, a precise KD determination is very difficult. This is in many cases related to solubility as well. So if you want to resolve a KD of 0.5 millimolar, you need to, in order to reach saturation, you need to go to compound concentration of about 10 millimolar. So that's already very, very challenging. Yeah, so I would say, to make a long story short, that a precise biophysical characterization, I would say, is possible, at least in the workflow that we are working, up to KD of about 200 micromolar.
See, another question asks, does the fragment-based drug discovery library have WuXi exclusive scaffold?
Yeah, maybe I should take that question. So I guess we started compiling that library based on a mixture of commercially as well as WuXi pre-owned fragments. However, it turns out since fragments are nowadays ubiquitously available, we find that actually we are not exclusive, and our clients can, most of the fragments source from any kind of sources. So the answer probably here is no. So there is no exclusivity.
And then another question here, I think we've kind of touched on this already, but in case there's something else you wanted to add to your previous answer, this question asks, what are the advantages of fragment-based drug discovery compared to other screening methods, and how about compared to HTS?
Yeah, I think I already commented on that, and I think the big advantage is the coverage of chemical space with the smaller fragments in terms of penetrating the chemical space better, finding new chemical space, and by that also being able, maybe for difficult targets that are highly competitive, managing your own IP and creating your own IP space. That's what I view as the most advantage.
Well, good. I mean, that's a great way to conclude this webinar by emphasizing that point again. So I want to say thanks again to Dr. Shoup and Dr. Jarabek-Williamson for your fascinating presentation, and thank you participants for being a great audience and asking great questions. Be sure to check CNN or CNN Online for information on the next edition of CNN webinars. Thank you On24 for technology and production services, and thank you WuXi AppTech for the sponsorship that made this interactive webcast possible. For CNN webinars, I'm Jeff Huber, and I hope everybody has a great day.